and Fisheries Universitas Hasanuddin, Bapak Safrudin, PhD.
Your Excellency, Director of Institute of Tropical Aquaculture, University Malaysia, Terengganu, Professor Muhammad Iwanuddin. The Honorable Invited Speaker, Professor Harry Paul, Rostock University, Germany.
The Honorable Invited Speaker, Dr. Ana Isabel Sousa, University Aveiro, Portugal.
The Honorable Invited Speaker, Associate Professor Dr. Nurul Ulfa Karim, University Malaysia, Terengganu, Malaysia.
The Honorable Invited Speaker, Dr. Davin Setyamarga, National Institute of Technology, Japan.
The Honorable Dr. Mukti Zainuddin, MSc, Universitas Hasanuddin, Indonesia.
The Honorable Invited Speaker, Dr. Amanda Linsley, Luther College, USA.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Let's start our program by reciting Basmalah. Bismillahirrahmanirrahim.
Let's praise to the Almighty God, Allah Subhanahu wa Ta'ala, that today we can participate in the 5th International Symposium on Marine and Fisheries with the theme
Advancing marine and fisheries sectors to achieve the sustainable development goals for healthy aquatic environments and society. Organized by the Faculty of Marine Science and Fisheries, Universitas Hasanuddin, in collaboration with the Institute of Tropical Aquaculture and Fisheries from University Malaysia, Terengganu.
Ladies and gentlemen, our first agenda is welcoming remarks as well as opening the symposium officially.
We cordially invite the Dean of Faculty of Marine Science and Fisheries, Universitas Hasanuddin, Bapak Safrudin, SPIMP, PhD. To the Dean, time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning. Honorable Rector of Universitas Hasanuddin, Professor Jamaluddin Jompa, Honorable Director of Institute of Tropical Aquaculture and Fisheries, Aquatrop University Malaysia Terengganu, Professor Muhammad Iwanuddin, and the team, Honorable Invited Speakers, Professor Harry Paul from Rostock University, Germany, David Setyamarga, PhD from National Institute and Technology, Japan, Dr. Amanda Linsley from Luther College, USA, Dr. Ana Sousa from University of Aveiro, Portugal, Associate Professor Dr. Nurul Ulfa Karim from Aquatrop UMT, Malaysia, Mukti Zainuddin, PhD from Universitas Hasanuddin, distinguished guests, respective presenters, colleges, and participants from abroad and within Indonesia, ladies and gentlemen. As the Dean of Faculty of Marine Science and Fisheries, Universitas Hasanuddin, on the behalf of the organizing committee, I am honored and more than delighted to welcome you all to the 5th International Symposium on the advancing marine and fisheries sector to achieve the sustainable development goals for healthy aquatic environment and society that is held in our beautiful Tamalan Rea campus, Universitas Hasanuddin in Makassar City, Indonesia. In this event, we will share and learn from each other through presentation and discussion on effective concept, strategy, policy, innovation, as well as lesson learned based on scientific researches on the various cases and projects related to the theme that are conducted within Indonesia and abroad. In the fourth International Symposium 2021, we talk about the challenging of in achieving the SDGs during the pandemic era of COVID-19. Today, as the pandemic hopefully subside and we are slowly adapting to return normalcy, we will be entertained with interesting topics such as fish parasitology in Indonesia by Professor Palm, molecular evolution and population genetics for marine by Dr. Setia Marga, examining local impact of blue economic policy with bioeconomic model by Dr. Lency, coastal wetland conservation by Fetch, the way to a climate action by Dr. Sousa, innovative seafood technologies for sustainable fisheries sector by Dr. Karim, and the detection of ocean spot for pelagic species within the Indonesia Fisheries Management Area 713 by Dr. Stainuddin and so forth. Ladies and gentlemen, the scientific findings that we gather from this symposium will be very essential for us as an agent of change to formulate our own local strategy and policy, particularly for Indonesian. This funding will be support the realization of three main programs of Ministry of Marine Affairs and Fisheries, namely, to improve the local productivity of potential marine commodity, including export and import, to improve income and well-being of the local fishermen, especially in low production chains, and to improve the healthiness of our coastal culture based on their local wisdom. We all know very well that the healthiness of our aquatic ecosystem has direct impact on the healthiness of ourselves, our society, and our future generation. And this relation applies vice versa. This is because our aquatic ecosystem serves numerous important functions, not only as the habitat of wildlife, food stock, and energy sources, but also part of the natural recycling and purification system, as well as disaster mitigation system. Therefore, protecting it from degradation, destruction, and harmful activities is no longer option, but must. Ladies and gentlemen, in this special event, allow me also to take this opportunity to introduce you our faculty, especially for some of us who have yet visited to the Tamalanrea campus. The Faculty of Marine Science and Fisheries was established 26 years ago, precisely on 29 January 1996. It's among 6 faculty in our university. The vision is to become the center of development and management of the coastal and marine sources. In total, there are 115 lectures, among which 23 are professors. It is consists of 2 departments, Department of Marine Science and Department of Fisheries. There are 5 undergraduate studies, 2 master degrees, and 1 doctoral degree. Currently, we are in progress to establish additional, regular, and 1 vocational study program. All study programs have accomplished the national accreditation, among which 4 are awarded with excellent national grade. Up to this day, the faculty has successfully accomplished the ASEAN Germany International Accreditation for the aquaculture study program, and another one is still in progress, the capture fisheries. It has also successfully accomplished the international certification of AUNKIE for the Marine Science Study Program and the Aquatic Resources Management Study Program. Besides that, 18 laboratories in this campus, we also have marine station, which located in the island of Baralompo, a fish pond in Makassar City, a fish pond also in Baru Regency, a new campus in the island of Selayar, where we conduct a vocational study program collaborated with the local government since 2021. This facility really supports the practice experiences of our students. At the same time, can be our selling points in the market branding of the educational institutional, especially in Indonesia. The vocational study itself is considered an example how to effectively reach, especially our young generation of low income fishermen communities that live in far island, and to offer them proper quality of education in this field in accordance to sustainable development goals. Ladies and gentlemen, finally, I would like to thank the chairman of the organizing committee, Dr. Siti Aslamia, for the dedication of hard work that she and her team have done. Our high gratitude also for the director of Aquatrop University Malaysia, Terengganu, for their support and collaboration in this event. Hopefully, we could maintain and expand our collaboration in the future. We hope this event could open the new potential collaboration with other universities and other organizations to spin up positive change and encourage better global management in our marine and fisheries development, especially in Indonesia. The last but not the least, we would like to thank everyone who participated in this event. We hope our hospitality would make you able to enjoy this event. Thank you very much. Have a great day. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Bapak Saprudin, PhD, for your delightful speech.
Ladies and gentlemen, next is the main agenda of the 5th International Symposium on Marine and Fisheries, the plenary session.
For the plenary session, we have 3 excellent main speakers. This session will be led by Dr. Asmi Citra Malina as moderator. To Dr. Asmi Citra, the stage is yours.
Before we start, we have coffee break 10 minutes. Thank you.
Ini Edgar nih, si tukang belanja online. Dia sedang memburu jaket. Saldo elektroniknya kurang. Selama top up jadi sold out.
Ribet amat belanja online. Debitin aja pakai Mandiri.
Hah? Debit Mandiri bisa buat belanja online?
Pilih bayar pakai debit, masukkan nomor kartu, masa berlaku, CVV di belakang kartu, dan kode OTP yang lo terima.
Lancar nih.
Kan didebitin pakai Mandiri. Saatnya debitin belanja online.
Saatnya mandiri.
Faculty of Marine Science and Fisheries, running internationally, is keeping the best.
Since founded in 1996, the Faculty of Marine Science and Fisheries, Hasanuddin University, has produced thousands of alumni and has grown to become of the leading institution in Indonesia in the field of marine and fisheries. Our vision is a referral center for development of management of coastal and marine resources in 2013. The Faculty of Marine Science and Fisheries has two majors, Marine and Fisheries Science.
I'm a proud student of Aquatic Resources Management Study Program, Faculty of Marine Science and Fisheries, Hasanuddin University. The study program is supported by lecturers with graduate expertise. Hence, at present, the study program has a national accreditation with rank A given by BANPETE, as well as internationally certified under the ANQA. For those who are interested for their education and expand their soft skills, students are encouraged to get involved in the lecture research activities, both at national and international levels. This approach has made our alumni employed in many fields such as bureaucrat, researcher in research agency, lecturer and teacher, manager and researcher in international and national NGO, as well as entrepreneur. So, what are you waiting for? Come and join us at Aquatic Resource Management Study Program, Hasanuddin University. Hi, I'm Layan Anggana Nagra, Aquaculture student 2018 from Hasanuddin University. Aquaculture study program is one of 5 study program in fishery, and one of the most interesting is aquaculture. This aquaculture study program has been accredited nationally by BANPETE with accreditation rating of superior and international accredited by ASIN. And the lecture have quality capacity and have international competitiveness. Taught us in the area of competence in fish parasite and disease, fish reproduction,
Ini Edgar nih, si tukang belanja online. Dia sedang memburu jaket. Saldo elektroniknya kurang. Kelamaan top up jadi sold out.
Ribet amat belanja online, debitin aja pakai Mandiri.
Hah? Debit Mandiri bisa buat belanja online?
Pilih bayar pakai debit, masukkan nomor kartu, masa berlaku, CVV di belakang kartu, dan kode OTP yang lo terima.
Lancar nih.
Kan didebitin pakai Mandiri. Saatnya debitin belanja online.
Saatnya mandiri.
Ladies and gentlemen, next is the main agenda of the 5th International Symposium on Marine and Fisheries, the plenary sessions.
For the plenary session, we have three excellent main speakers. This session will be led by Dr. Asmi Citra Malina as moderator. To Dr. Asmi, the stage is yours.
The Honorable Rector of Hasanuddin University, Professor Dr. Jamaluddin Jompa, the Honorable Dean of Faculty of Marine Science and Fisheries, Safruddin SPE, MP, PhD, Chairwoman of the Committee of the 9th National and 5th International Symposium on Marine and Fisheries, Dr. Insinyur Siti Aslamnia, Chairwoman of the Partner Committee, University Malaysia Terengganu, associated from Dr. Marlina Hassan, the Honorable invited speakers, distinguished guests, all the participants, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. May peace be upon us. Very good morning to all in Indonesia. Good afternoon and good evening to the citizen in the world. First of all, I would like to welcome you all to the 9th National and 5th International Symposium on Marine and Fisheries Science, held by Faculty of Marine Science and Fisheries, Universitas Hasanuddin, in collaboration with University Malaysia Terengganu. It is wonderful conference today that will be held with two sessions from morning to this evening today. And I'm Chitra, I'm from Fisheries Department, Faculty of Marine Science and Fisheries, Universitas Hasanuddin. It is great joy and privilege to me moderating this symposium today with the theme Advancing Marine and Fisheries Sectors to Achieve the Sustainable Development Goals for Healthy Aquatic Environment and Society. Actually, with the very prominent speakers that also will be very important speakers that present today. Before we start the first session, I would like to the invited speakers today. I will be moderating for the first session, and we have 3 invited speakers today. The first speaker is Professor Harry Palm from Rostock University, Germany. Hello, Professor Harry.
Hello, can you hear me?
Yeah, I can hear.
Very good.
Or good morning there.
Well, it's 3 o'clock in the morning.
Okay. How are you doing?
Yes, well, I'm doing fine.
Sorry, very morning to organize this session. And the second speaker, Dr. Ana Isabel Sousa from University of Aveiro, Portugal. Good morning or good?
Hi.
Hi.
Good night.
night.
It's 2 o'clock in the morning here.
Okay. What time is it now there?
2 a.m.
2 a.m. Oh, very sorry to disturbing your night time. Must be sleepy now. Okay, the third speaker, associated from Dr. Nurul Ulfa Karim from University Malaysia Terengganu. Assalamualaikum warahmatullahi Prof.
Waalaikumsalam warahmatullah.
Selamat pagi.
Morning, everybody.
Morning, yeah. And before we start, so maybe I would like to introduce the first. Yeah. Now we can start. Yeah, sorry for there is a little technical error. Yeah, please open the Zoom. Now we start the first session. Please allow me to invite Professor Harry Palm and allow me to read the biography. Professor Harry Palm's with the expertise aquaculture and sea ranching, right?
Yes.
From Faculty of Agricultural and Environmental Sciences, University of Rostock, Germany. And Professor Harry Palm have published 4 books and 1 book in preparation, 2 monographs, over 100 peer-reviewed publications in international journals, and now 10 in preparations. And over 20 different journals as a referee for international journals. So we invite you to give presentation, Professor. And your time, I think, how many minutes? 20 minutes from now.
Well, I will try to share my screen, so I hope that this works.
Yes. Please.
Can you see that?
Yeah, okay, we can see it.
Yes, perfect. Okay, yeah, thank you very, very much for this invitation. And I want to present you this morning or this night about our status, about our results, that we are doing fish parasitology research since about 25 years now in Indonesia. And I want to give you a short background, the current state of knowledge, what we know nowadays. And I also want to scratch the topics, how we can use fish parasites as bioindicators, because this issue is pretty important for understanding marine ecosystems in Indonesia, and also in dealing with the sustainability of heavily used ecosystems in future. So why are we working with fish parasites? And it's an honor for me now to start with this topic, because people don't realize very often how important fish parasites are in the marine ecosystems. So normally these fish parasites are embedded in any marine ecosystems. There are a lot of trophic relationships in the marine systems, and these parasites, they migrate actually and are given from one host to the other one. And we also have a very high biodiversity of these organisms. And this is normally heavily underestimated. So there are by far more fish parasites than fish species existing. And also that means in the marine ecosystems, if we are dealing with fisheries, we have a sheer number of fish parasites, much more than the fish species we are dealing with. So we can also use these fish parasites to understand about trophic relationships of the fish inside the natural and also the heavily used ecosystems. So the very interesting thing to show you how these organisms are linked together, I have this graph here. This is a nematode, which normally is living in seals in the North Sea. And you can see here a time series. And this seal, they had a disease. So there was a real drop in numbers. And then after this disease event finished, the number of seals in the environment increased again. So when you are now studying the fish parasites, you can see the adult is living in the seal and the larvae are migrating through the food web, and finally you can find them in the fish. When you then study the fish, you can see in terms of abundance and number of parasites inside the fish, you can see that there's a kind of a time lag. But the parasites actually, the infection rates follow the numbers of the seals in the ecosystem. So the parasites indeed can describe very nicely the abundance of very important hosts in the system. So, and also fish parasites are not new in Indonesia. The first fish parasite was described already in 1840. And there was quite some research at the beginning of last century, which focused on crustacean fish parasites. And the first fish parasitologist, it was a Japanese scientist. He started in 1952, and interestingly, in Makassar, Sulawesi. Then finally, we had a lot of other scientists, especially working with museum material. And in about 1975, a lot of Indonesian researchers started with fish parasitology research because of mariculture, increasing mariculture in your country, and also because they were searching for anisakid nematodes. This is a nematode species which is zoonotic to humans. It can be transferred to humans. And you can see here a series of Indonesian authors. Myself, my students, and my Indonesian students started in around 2000, so about over 20 years ago. And with a highlight when we had a first educational workshop on fish parasites in Bali, Indonesia in 2013. So our issue over all these 25 years now, it was always to explore the fish parasite biodiversity. You can see here different fish parasites in Indonesian fish. And we wanted also to use the fish parasites as bioindicators for ecosystem health, and we wanted to monitor mariculture and wanted to see whether mariculture has also impact on the natural ecosystems in the area. So the problem in Indonesia is that you have a huge aquatic and marine biodiversity. So if you have a look here, for example, we know already from other research areas about protozoans and flukes, gill flukes and cestodes and digynians. We know about several fish parasites that are quite dangerous for mariculture. But the point is, when we are studying in Indonesia, this is just a species list of fish parasites that we have found in some 2 or 3 fish species that we sampled in Segara Anakan Lagoon in Central Java. And what you can see here, if you study fish parasites, the biodiversity in your country is huge. And of course, this biodiversity is far from being exploited. There's a new publication which just came out this year or last year by a former student of mine. She summarized the known fish parasites from mariculture fish. So especially groupers and others, and this has been published by IPB Press. And so we have now identified the most problematic fish species in your country. And so you can really look into this publication if you want to inform yourself about the biodiversity in mariculture fish. The other second important publication from us is by one of my PhD students who published a list of all articles and records of fish parasites from Indonesia since 1840. And he recorded 621 different fish parasites from 315 free-living marine teleosts. And if you see this number, 315, that means that over 2,700 or even more fish species have never been studied for fish parasites at all. So he suggested that we only know about 5.2% of the Indonesian marine fish parasite fauna in total. So this is only a fraction. So now I want to summarize our results about these zoonotic worms. Zoonotic worms can be transferred to humans. So if we eat the fish, we can get the worm actually and get diseased. And one very important species is Anisakis simplex. And this was also the origin of the series of papers from the Indonesian scientists, because we all wanted to know how many Anisakis species are living in Indonesia and are they a threat to human consumers, to consumers who are eating fish from your environment. So these Anisakis species you can see here, Anisakis simplex has a distribution in the northern area, in the boreal zones of the Earth. It's a cosmopolitan, mainly cosmopolitan species. And the interesting thing is we found another species, which is Anisakis typica. And if you go now to Indonesia, actually Anisakis typica is the most common species. So it's not Anisakis simplex. This has been done by molecular IDs. So Anisakis simplex, that would be potentially a threat to the consumer, actually is not a real problem. We published these results and you can actually read this in this paper. But the interesting thing is, if you look now, we know over 53 species of teleosts that are now infected with Anisakis and with this genus of parasites. And if you look into the world, there are only 177 total species that are infected with this parasite. So you can see 1/3 of all records ever been reported are from Indonesia. And this shows you how interesting and important data are from Indonesian waters. So I only have 20 minutes for this presentation, and now I want to focus on a second very important aspect of fish parasitology. This is when you can use fish parasites as biological indicators. So for example, for stock separation, we can use them. We can use them for migration patterns of fish in the ocean. And also we can use them as environmental indicators. So to show pollution, change in the environment, so ecosystem bioindicators. And finally, here in Europe, we are also using them as bioindicators for global change. So there are a lot of different possibilities how you can apply fish parasites. And it's important to only use certain fish parasites. I told you there's a huge biodiversity, but you have to select those ones that are easy to collect and that are easily recognizable, also not from the expert like ourselves, but also from somebody who is interested to apply this methodology. And you can see that with such parasites, and I show you here, this is a species I described with one of my former PhD students, Gunarto Latama. He collected this worm actually from Makassar in Indonesia. And I wrote a very nice paper on the ultrastructure of this paper, of this worm. And so to try to use fish parasites as bioindicators, we sampled actually in the Southern Islands a mariculture farm, which is at the beginning, you see here, it was only for grouper mariculture. It's in the Southern Islands. And after some time, the farm expanded. Here you can see a milkfish net cages which are attached to the farm. So in these years, when we monitored this farm, actually the production strongly increased. And what we did was we developed a new method where we actually wanted to show how these parasites can tell us how to visualize an environmental change, and that could be used, that could be monitored actually by parasites. So we selected certain fish parasite species or indicators like ratios or the Shannon-Wiener Biodiversity Index, which is an index used by many biologists. And then we developed a kind of a star graph. So we centered these graphs, and at the beginning we only had 3 of these characters, but then we moved on. You see now we have here 6 of these characters where we then sampled, and each point here, each dot here is a sampling point over 6 consecutive years. Yeah, so we sampled the mariculture farm over 6 consecutive years. And now I want to show you the results. So this star graph reads as follows. So the worst case in the environment is if the parasite indicators would be here. The best case is if the indicators would be here. So the largest surface of this star graph shows you the best environmental conditions in that place. So now I just show you what happened during the 6 consecutive years. This is the first year. This is the second year. This is the third year, the fourth year, and you can see some of these branches are collapsing. This is the fifth year, and this is the sixth year. So you can see this aquaculture farm actually, in terms of fish parasite indicators, was deteriorating its environment. Yeah, so you can use actually the fish parasites to demonstrate what is happening in this place. And you can see what happened in the farm. You can see when we started to sample, there was only a small production. But then the production increased. And finally, after 6 years, we had the highest production. So while increasing the production, actually the environment deteriorated, and we could monitor this by monitoring the fish parasites in the groupers inside the aquaculture farm. So I now want to come to the end of my presentation. So we have further developed this, this methodology, and this shows you actually different environments that we sampled for grouper fish parasites, and this is Jakarta Bay. So if you go to the jetty in Jakarta and you sample groupers, you will find a parasite, in our system, a parasite value, which is much, much worse. This is also the coastal areas of Jakarta, and these are areas in Bali. This is Bali, and this is Segara Anakan Lagoon. So you can see, you can also show what is the situation in different Indonesian marine ecosystems. And in this publication, actually, we analyzed 4 different aquaculture sites. So we could also show that, for example, the Cat Ba region in Vietnam here was much overexploited by aquaculture farms, and we could tell this just on basis of analyzing the parasites by using the star graph system. So finally, to close this up, we can say as a conclusion, we only know about 5% of the fish parasite fauna in Indonesia. That is only a fraction. So many new species are to be expected, and some of them for sure will also have relevance to aquaculture. The fish parasites can be used for stock assessment and as environmental indicators. We have demonstrated this, and we also have the methodologies to do so. In 2013, we had this workshop, the first educational workshop, which revealed 325 new parasites records in only 2 weeks. And actually, this is a huge number in comparison to other areas. And you can see here, we are publishing a lot of papers still only based on this workshop in Bali. So my suggestion, and this is something to give you to Makassar, you know, I'm a friend of your university since many, many years. And my suggestion would be, why don't running a second educational workshop on fish parasites in Makassar in future? So thank you very much. I hope you will have a successful symposium, and thank you very much for inviting me to give this presentation.
Thank you very much, Professor Harry Pham, for the interesting presentations. So we now would like to invite the second speaker, Dr. Ana Isabel Sousa from University of Aveiro, Portugal. Are you ready? You have 20 minutes from now. Time is yours. Okay, sorry for, before you start your presentation, Please allow me to read your biography, Dr. Ana. Dr. Ana Isabel Souza expertise on natural sciences, biological sciences, and ecology. And she has many publications. She co-author 20 ISI papers, 11 as leading and or corresponding author, and 14 in the last 5 years. And Dr. Ana Isabel have published 6 book chapters, 1 book, and 2 technical reports. She has achieved an H index of 13 with a total of 849 citations, 820 excluding self-citations. And it's yours, Dr. Ana.
Thank you. Can you see my screen now?
Yes, please.
Okay.
To make it.
Okay, so good morning, everyone. Sorry.
Good morning.
I'd like to thank you for the invitation to attend this conference and to join you. So I'm Ana Souza, and I'm a researcher at University of Aveiro. My talk will be about coastal wetlands conservation and their role on climate action. So it is shown that it paves the way to climate action. And I'd like to talk about this in detail. So we have different types of coastal wetlands, and in this case, I'd like to focus on salt marshes, mangroves, and seagrass meadows. The seagrass meadows, sorry, I'm coughing a lot. Sorry, the seagrass meadows can be either intertidal or subtidal, and they are distributed worldwide. This is an example of a Zostra marina species, which is subtidal, and their distribution is all over the world except in Antarctica. But there is a difference in terms of species diversity. So in tropical areas, the species diversity is higher than in temperate areas. Regarding salt marshes, we have also a global distribution of these ecosystems, and this picture is taken at Ria de Aveiro in Portugal, and we can see extensive areas of salt marshes. These plants are adapted to live under a different range of salinities and flooding, and according to the tide. And this picture is a picture of a salt marsh, an image of a salt marsh during low tide. The third ecosystem are the mangroves. Sorry, which are common in tropical areas. These coastal wetlands are really relevant, not only for their existence or their presence, but they are important as habitats for many different species of invertebrates or fishes or lugworms, etc. So these habitats are really the biodiverse. They promote the biodiversity, and they are important areas for as nursery areas for some fishes and some invertebrates. These three types of ecosystems, seagrasses, mangroves, and salt marshes. They follow an elevation gradient, and they have different important functions and services. So they all of them contribute to the protection against the erosion and the protection against storms. They also contribute to the pollution remediation and to the sediment control. They contribute to the sedimentation of the sediment there is in the water column. Another relevant function is the improvement or enhancement of the carbon sequestration. These ecosystems are really important in the carbon storage, and that's why they are known as the blue carbon ecosystems. The blue carbon term was firstly used in 2009, and it corresponds to the carbon that is stored in the oceans and in the coastal wetlands, but mostly in coastal wetlands, and both in the living biomass and in the soil, in the sediment. So this carbon, the CO2 fixation can contribute to the conversion into organic carbon, and this carbon can be stored in the sediment for centuries or millennia. So these ecosystems contribute to the greenhouse gases reduction in the atmosphere, thus contributing to climate regulation. When we compare the capacity of storage of these ecosystems with other type of ecosystems, such as the forests, we can see that the carbon stored is much higher in blue carbon ecosystems when compared to the forests. And this is due to the anoxic conditions within the sediment that contribute to a slower decomposition when compared to the forests. Besides this, most of the carbon is stored in the sediment, and a smaller proportion is stored in the living biomass. So even though the coastal habitat coverage is only about 2%, these ecosystems contribute to the storage of 50% of the carbon. So this highlights their relevance in carbon storage and climate mitigation and adaptation. This is just a map of the global distribution of the 3 types of blue carbon ecosystems. And so plenty of research has been done to study the role of these coastal plant communities to mitigate the climate change and to store the blue carbon. An example regarding of a study done at Ria de Aveiro here in Portugal was a work that we selected 3 sites in the Ria, which is a coastal lagoon. It's a Ramsar site, and it's included in the Natura 2000 network. So we estimated the blue carbon storage in different, the blue carbon and also the nitrogen and phosphorus, but now I would like to point only the carbon. So we monitored the biomass over 1 year, each 2 months, and the biomass and the carbon stock, and we estimated the amount of carbon that was stocked in both in the plant and in the sediment. This is an example of the different species that we have here: low marsh, mid marsh, and high marsh. besides their role, there are some threats that are increasing the vulnerability and destroying these areas. One of them is the erosion that you can see in figure A, but also the increase in the changes in the physical forcing in the lagoon that leads to changes in the time of inundation and flooding. So there is some dying of some plants. Regarding the seagrass meadows, we have about 230 hectares in Rio de Aveiro, and we monitored the seagrass area and compared it from 2005 and 2014. What we noticed was that there was a slight shift in the area of the meadows, or in the site of the meadows, and there was some recovery in some of the meadows. So, but other meadows were declining. So there was some vulnerability and some changes in the aerial extent, and these will lately affect the blue carbon stock in a medium or long-term perspective. We estimated the carbon stock in the plant and in the sediments, and we then assessed it for some areas every month, and then we estimated the blue carbon stock for the whole system. So this blue carbon stock can go until 450 megagrams of carbon that is stocked in these seagrass meadows, unless they are then threatened or under some pressures that may destroy these areas and likely affect the blue carbon storage. So even though their relevant role, they have been declining worldwide in the last decades, and some relevant papers were published highlighting these decrease and the loss of seagrasses. But there is some hope still, and there was a paper published recently in 2019. And it was found that there was some small or some slight trend reversal for this declining, which is quite good. And then it's the example of the data we have collected in Rio de Aveiro with slight recoveries in some meadows. These seagrass meadows are under threat through different types of pressures, such as the sea level rise, the harvesting, the presence of invasive species that affect the equilibrium of the system, but also the ocean acidification, the dredging activities, etc. So there are some works that are highlighting the vulnerability and the risks that these areas or these ecosystems are facing in these times of change. There are some relevant questions that were highlighted by a group of scientists that work on blue carbon, focusing on the disturbance. How does the disturbance affect the burial rate of carbon? How does the climate change impact the carbon accumulation, both in restored ecosystems or in nature ones? And also the question was, what are the best management actions to promote the blue carbon sequestration? So this is some of the topics that are under discussion. Regarding the seagrass meadows threats and changes over time in Rio de Aveiro, we have noticed a decrease in diversity of the species. From 50s to the 80s, we had subtidal and intertidal seagrasses, and until the 80s. But after that, we have only recorded intertidal seagrass meadows, and also the diversity of seagrasses has decreased. So the last record of Zostra marina had been in 2008. But we recently, in 2019 and 20, we found some Zostra marina patches. So we are now following them, and that's a good news for us. Some of the threats that have been disturbing this system are the dredging activities and the sediment remobilization that occurs due to these activities. Also, the increase in water current due to the changes in the physical forcing, and some loss of nutrients and fine sediments that prevent seagrasses to be anchored. On top of this, there was a recent threat, which is the presence of an indigenous species, a lugworm, Arnicula marina, that was introduced in 2009. This is the view of a seagrass meadow with plenty of fecal casts from this lugworm. And we can see here in this picture that the remobilization of the sediment by the construction of their galleries affects the seagrasses, increases its fragmentation, and there is a loss of biodiversity in the system. So in 2008, we started a project, Biopradaria project, where we intended to restore and manage and conserve the biodiversity of the seagrass meadows at Rio. So we implemented some restoration measures. We applied nature-based solutions, such as a natural membrane. And just to show you some of the restoration activities that we did, this is the membrane, and these are the sods that we transplanted, and then we monitored over time. So our goal was to increase the resilience of Zostra nolte against this lugworm. And of course, afterwards, with its restoration, the goal is to increase biodiversity and also the carbon storage in the system. In terms of climate action, and there is plenty or a vast policy agenda, and at the European level, there is the EU biodiversity strategy, the climate action, and also the European Green Deal, which include the conservation of these areas and decarbonization of the countries. At international level or a global level, we had the Paris Agreement established in 2015. Sorry, it was before, but in 2015, the new targets were established. And the goal is that the global warming will be below 2 Celsius degrees, that each country will contribute to, or to try to mitigate the global warming. So the national determined contributions or the plans for climate action were established by each country by 2020. And then the goal is to improve the resilience of the different ecosystems and reduce the greenhouse gas emissions. So there is a great opportunity now. We have the UN Oceans Decade and the UN Ecosystem Restoration Decade going on. So that's relevant funding to promote these restoration activities and improve these systems and the blue carbon storage and reduce of or mitigation of the climate change. Other policy opportunities or policy strategies that were implemented is the Sustainable Development Goals, and in particular the climate action, the SDG number 13. Blue carbon, the blue carbon was highlighted, or the blue carbon stored by seagrasses was highlighted also by the definition of or establishment of 50 sites as the UNESCO Marine World Heritage, given their role on blue carbon storage. So they are considered significant opportunities to mitigate the climate change and to meet the goals of the Paris Agreement. Regarding the nationally determined contributions, there are only some countries that have included the seagrasses on its accounting. So there is a goal to increase the number of countries that include these type of ecosystems on the national determined contributions. And also this should be integrated with both the blue carbon storage and integrated with the local coastal communities. So the people, the policy and finance, the science and technology should all together be integrated so that the blue carbon can be highlighted as a NDC at a local level. In Portugal, the long-term strategy for carbon neutrality did not include the coastal wetlands. So there have been some meetings with NGOs, scientists, and the national authorities to show them to show the authorities the relevance of including these blue carbon ecosystems in the NDCs. So there is an urgent need to protect and to restore these ecosystems, and there is also several opportunities, and we should use it to protect these areas and to contribute to climate change. Thank you very much for inviting me, and it was a pleasure to show you some data. Thank you.
Thank you very much, Dr. Anna. Very interesting presentation. And we now move to the third speaker. We would like to invite Associate Professor Dr. Nurul Ulfa Karim. Yeah, please allow me to read the biography of Professor Dr. Nurul Ulfa. Has an expertise on fisheries post-harvest, and she's from Institute of Tropical Aquaculture and Fisheries, University Malaysia Terengganu, and she has 14 ISI papers as main or corresponding author and 11 papers as co-author. And she's Professor Dr. Nurul Ulfa. Research interest is fisheries post-harvest technology, includes thermal and non-thermal processing technology, traditional and modern preservation techniques, and application of natural product and packaging system. Please welcome Professor Dr. Nurul. Time is yours. You have 20 minutes from now.
Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and very good morning. First of all, I would like to thank Universitas Hasanuddin for inviting me as the speaker for the 5th International Marine and Fisheries Symposium, advancing marine and fishery sector to achieve the sustainable SDG for healthy aquatic environments. Okay, so all right. Okay, and special thanks to the moderator, fellow panels, and Symposium Secretariat for the introduction and warm greeting. My name is Nurul Ulfa Karim. I'm a lecturer and researcher at Higher Institution Center of Excellence, HICOE Institute of Aquaculture, Tropical and Fisheries, University Malaysia, Terengganu, Malaysia. And today I will share with you on innovative processing technology for sustainable fishery sector. So this is the outline for today's presentations. Firstly, I will talk about the seafood spoilage and innovative seafood processing technology, and also the green seafood processing, the natural preservatives, the essential oils and plant extract. And the last topic will be discussed on the sustainable fish, seafood, value chain, trade and climate. So what is seafood actually? Seafood are defined as a free-swimming, pelagic and freshwater fishes. It also include crustacean, mollusks and the respective aquaculture species. The most important crustacean includes crabs, lobster, crayfish, shrimps and prawns. Fish are further categorized as the saltwater versus the freshwater fishes, fatty or non-fatty fishes, and free-swimming and aquaculture fishes. So the fish compositions varies with species, size, and fishing grounds and diets. Okay, why is a fish easily spoil and perish? The fresh caught fish are normally sterile. Only after harvest, the fish skin carry about 10 to the power of 2 to 10 to the power of 7 CFU per cm squared bacteria. Meanwhile, the fish gills and intestine carried about 10 to the power of 3 to 10 to the power of 9 CFU per gram bacteria. The fresh ice-thawed fish consists mainly the gram-negative bacteria such as Pseudomonas or H2S producing bacteria including Salmonella species, Acinetobacter and Moraxella. Immediately after caught, the post-mortem biochemical changes occur as the result of autolytic enzyme in fish flesh. The autolysis affects two things, which is first one is the texture of the fish muscle tissue at early stages of deterioration, and secondly, autolysis enzyme of the muscles and visceral organs drive autolysis during the processing and storage. All those will lead to the protein decompositions and solubilizations, and number two, produce the peptides and free amino acids, and the third one, the proteases and lipases degradations. Okay, therefore, innovative and recent developments on the seafood processing technology has been widely applied in fishery sector. As mentioned earlier, seafood products are very perishable commodity. Consumer demand for high quality, lightly processed product with minimal changes in nutritional and sensory properties. Rapid development in seafood processing facilitate the transformations of traditional fishery sector. So what is the traditional fishery sector? Traditional fishery processing includes the chilling, which is about 0 to 8 degree preservations, freezing, which is below than 1 degree preservations, whether it's drying, it's involved thermal technology, smoking, also involve the thermal technology, salting, fermentations, and canning. Okay, so the recent method that have been used worldwide nowadays is natural preservations, the high pressure processing, the ozonations, the irradiations, plus light technology, retort posh processing, or packaging in combinations with refrigerations or freezing operations. Okay, green seafood processing includes innovative and sustainable food process, robotic automations and control of food process, role of sensor and modeling in quality and process optimizations, and the fourth one is assuring a safe, authentic, and biosecure food supply chain. And today, I will only focus on the use of natural preservatives in seafood processing. So natural preservative seafood processing, there is six involved in natural preservations, which is effective against a broad spectrum of bacteria and fungi, active at low concentration, and it is a must to use the natural preservative, which is non-toxic, not affecting food sensory properties, and impart no flavor and color to food. The most important thing by using the natural preservations is it is a cost effective. Okay, the main category of natural preservation in seafood product is the use of the essential oils and plant extract. Okay, EO containing a number of other volatile organic compound, VOC, that produce a secondary plant-based metabolites. EO also should inhibit the germinations and growth of the pathogens. The EO offers antimicrobial properties against various foodborne pathogens such as E. coli and Listeria. EO also have hydrophobic nature that able to interact with the bacterial lipid membrane and thus lead to an increased permeability of cell constituents that resulted in cell death. Okay, so plant extract is a mixture of phytochemical compound extracted from plants. The phytochemical compounds are the byproducts of plant metabolisms that includes tryptophanase, phenolic compounds, and alkaloids. All bioactive phytochemical compounds occur in various parts of the plants, such as leaves, stems, flowers, barks, seeds, and roots. Both EO and PE, or essential oil and plant extract, are efficient in controlling foodborne pathogens and spoiled organisms. They both, which mean both EO and PE, destabilize the cellular structure by solubilizing in the lipid bilayer of the plasma membrane and mitochondria. And this leads to an increase in permeability of the cell membrane, resulting in loss of cellular constituents and disturbance of the active transport of substance. The antioxidants property of EO and PE are mainly due to their high content of phenolic compound. And you have to remember, EO and PE are usually used as the seafood preservatives at concentration range from 0.1 to 1%. At higher concentration, in most cases, there is a negatively effect product sensory properties. Okay, so my research interest also in using the plant extract in the natural as the natural preservations in seafood processing. We are looking on the effects of guava extract coating on the giant freshwater prawn. So guava leaf extract were tested for the antioxidant properties and activities such as the TPC, the total phenolic content, the TPC, sorry, the TFC, the total flavonoid content, the DPPH, and FRAP. The giant freshwater prawn was subjected to guava leaf extract coating at the concentrations of 0.5 and 1%. As the results, the TPC is quite high in guava leaves, which amounted about 338 mg/GEE per gram. So we also looking on the TFC, which accounted about 51 mgQE per gram. The DPPH and FRAP were at 77 and 2.5 micro TE per gram respectively. Guava leaf extract were significantly effective to reduce the total bacterial accumulations in Rosenbergi. Untreated M. Rosenbergi reached the limit of acceptability at 7 days. However, interestingly, the shelf life of both samples coated with 0.5 and 1% were extended up to 10 days of storage. With regards to the safety of food consumptions, microbiology analysis are the most reliable to reflect the shelf life predictions of M. Rosenbergi coated with the guava leaf extract. Secondly, we also have had look or published a paper on the moringa leaves, the effectiveness of moringa leaves extractions on the quality changes and melanosis of the GFP. Okay, so the extractions of moringa leaves were prepared as edible coats for the to keep the quality of giant freshwater prawn M. Rosenbergi. The antioxidant properties and activities such as TPC, TFC, DPPH, and FRAP of the moringa leaf were determined. So we found that the total phenolic compound is about 16 mgGE per sample. Meanwhile, the total flavonoid content is about 5.5 mgQE per gram. Meanwhile, the DPPH and FRAP is about 1.3 and 3.0 mgTE per gram respectively. Interestingly, moringa leaf extract will actively reduce the microflora count in M. Rosenbergi. So the total volatile basis nitrogen, the TVBN, one of the chemical indicator for the freshness, showed a significant lower amount in treated samples compared to the controls. So if you can see it here, initially there is no melanosis formations in all samples, but the controls has an obvious melanosis formations compared to others during the storage. So 1.25% SMS retard the melanosis formations, and the M. Rosenbergi deep with the moringa leaf extract either at 0.25 or 0.5% and 0.5% of ascorbic acid had a moderate melanosis formation. Okay. So recently, we have also published a paper on the effectiveness of Annona muricata extractions on inhibitions of specific enzyme on melanosis, which is the polyphenol oxidase, and the microbiological quality of the M. Rosenbergi. So we also tested for the TPC, TFC, DPPH, and FRAP of the Annona extract, and we also looking on the effectiveness of Annona muricata extract on the PPO activity and bacterial accumulations. So we found that the total phenol content is about 191 mgJEE per gram. And this is the most interesting findings. We found that 1,777 the total flavonoid content in Annona muricata leaf extract. Meanwhile, the DPPH and FRAP is about 1.3 and 0.7 mgTEE per gram respectively. 60% of the Annona muricata extract showed a significant effective to inhibit the highest level of PPO activity. It's about 82%. So, which means if you use 16%, we can reduce the PPO up to 82%. The A. muricata or Annona muricata leaves containing phenolic compound inhibits the PPO by competing with the substrate naturally present in the giant freshwater prawn. This with delaying the melanosis and therefore slowing the rates of discolorations. The total bacterial count show no significance The total bacteria count showed no significant difference between the controls and M. rosenbergi, coated with 10% of Anonomoricata extractions at storage day of 4 until 16. Interestingly, treatment about 15% Anonomoricata effectively reduced the bacteria accumulations compared to the higher level of extractions, which is a 20% at the same storage period. Similar trend were found in M. rosenbergi coated with SMS. The Anonomoricata leaf extract at approximately 15 to 16 contribute to delay the melanosis and shelf life extensions of M. rosenbergi stored at chill conditions. So last but not least, how is seafood processing may benefits to the or supporting the DSG. So I think for seafood industry, seafood processing industry benefits in the socials, especially to the fishermen. Fish and fishery products are significant to contribute to the food security and livelihoods. The industrials and developed country have higher annual per capita consumptions of fish than the global average, consuming about 26.8 per kilogram and 23 kilogram respectively. It's about 59 or 60 million employed in fisheries and aquaculture in 2016, and approximately about 200 million direct and indirect employment along the value chain from harvesting to distributions.
Excuse me, Prof. We are running out the time.
Okay, this will be my last slide. Thank you. It's 35 and 38% world productions enter international trade and sustain demand trade liberalizations policy, globalizations of food system, improve of the transportations and logistics, and also technological innovations. Lastly, the seafood processing industry also has the economic benefits on the high demand and the globalizations of utilizations and distributions. For that, thank you.
Thank you very much, Professor Nurul. Now we have completed the 3 speakers for the first session. So now we have a discussion sessions. We would like to invite the participant or academic researcher who would like to give some questions. Please, we now have many, we received many questions in the chat. Yeah, and have replied from Professor Harry Palm. And we would like to invite you to give some questions also through chat room, or you can just raise your hand, please. Yeah. Yeah, there is one for the question for Professor Harry Pan has been answered, and we have another one to directed to Dr. Ana from Bapak Sulistiono, Mr. Sulistiono, IPB University. The technique to improve. Yeah, I would like to read it. Thank you, Dr. Ana, for nice presentation. The technique to improve the seaweed ecosystem that was conveyed, I think, can be done by the community and all of us. But usually the problem faced is the willingness of the parties to rehabilitate the ecosystem. In the case of Banten Bay in the 1990s, the expanse of seaweed was very wide. Currently, there may be 10% left, and there seems to be no indication to rehabilitate the ecosystem. I would like to ask, what is the strategy in your country to be able to include the stakeholders involved in the rehabilitation of the seaweed? Because seagrass beds are usually not visible to the naked eye compared to mangroves or other land plants. Please, Dr. Ana.
Thank you for your question. Actually, it's not very easy to involve the local communities, but it's an effort that we that are doing the research and are that intend to restore the systems, that we should really make the effort to first to aware them about the local communities, about the importance of these ecosystems, and then to involve them since the beginning of the projects in these type of restoration or conservation projects. That's something that we have been doing in the Biopraderia project. So we have had some workshops with local stakeholders from different sectors, both fishermen, people from the tourism, from the local authorities, from nature reserves, and so on. So we get everyone together to let them know about the seagrass ecosystems that we had nearby their area of living or their area of activity. And then it's not an easy process, and it's not only with one activity. So it's a work in progress along different. We have been working with them since 2008, but former projects have been done before, not focused on seagrasses, but it's a work in progress. And it's relevant that when they are aware, they also work with us on the restoration activities. And that's something that we have planned for the future projects when we will implement restoration in a larger scale. Thank you.
Thank you, Dr. Ana. So, Mr. Sulistiono, Alice, do you still have any questions to Dr. Ana? Okay. We still have, yeah, now we have to, there is one another, yeah, I cannot see from this top, one raise hand, okay. Pak Hussein, please.
Okay, thank you very much. It's very interesting. Thank you, Dr. Ana, for presenting such a good presentation about blue carbon in Portugal. So it is interesting also about the case in Indonesia, because some prominent researchers estimate that Indonesia also has like the largest seagrass ecosystem in the world, but you just state that Indonesia still not include these blue carbon ecosystem, especially seagrass ecosystem, in its nationally determined contributions. So maybe I want to have your suggestions to our government, how to make them really want to include seagrass ecosystems, why seagrass is important to combat climate change. Thank you.
Thank you for a question. Actually, that's a fight that we are, a fight or a willingness that we have also here in Portugal, and it's not easy again to work. I think it's from sector to sector. It's not easy in any time, or not fast. So here in Portugal, and apparently the same in Indonesia, is that the authorities are not aware of the relevance of these ecosystems in reducing the greenhouse gases and in their contribution to the climate change, or to mitigate the climate change. So I think that the first step would be to contact the authorities, the national authorities, to show them their relevance. And that's something that we did in Portugal I think it was last year. So there was an NGO that was promoting, and I showed a slide with some newspapers news about that meeting that occurred. So an NGO promoted a joint meeting with scientists and the local or the national authorities responsible for the roadmap for carbon neutrality. So, and they were the NDCs, the agriculture fields are included in the NDCs, but these ecosystems are not. So we were actually trying to show them the relevance, but it was not accepted at the meeting or 1 month later, and it's still under discussion. It was last year, so it's not a fast process. But I think that as scientists, we should try to do it, and I think it would be relevant for every country because it will also promote the conservation and the restoration of these ecosystems. It would be a way that all the different sectors in the country would be interested in protecting these areas. But it's not easy. Thank you.
Okay, thank you. Maybe any experience from your country, how long to restore like seagrass or mangrove ecosystem, how long the process?
In mangroves, I've never worked with mangroves, so I cannot answer about it. But on seagrasses, it depends on your goals. So if the goal is to increase the carbon storage, then you can only see the benefits of or the increase in carbon storage after several years. But of course, you can monitor the restoration that you did, and then you can see the increase in the coverage of the seagrass meadow, and then that you can see a bit faster in less years, but it's always a long process.
Okay, nice, thank you.
You're welcome.
Thank you very much. And so we will close the session, or no raise hand again. Yeah, I think there is no more question for the 3 invited speakers, and we now also have to move to the second plenary session, for part 2. And before we close, there is a summary for the 3 speakers from Professor Hariparam. We show how fish parasites can be used as bioindicators for environmental waters. And from Dr. Anna, she showed that it's very important to taking urgent to combat climate change, focusing implementation on SDG 13. And Professor Nurul Ulfa find the innovative seafood processing technology for seafood applied for preservation and shelf life extension. Seafood products have been widely applied worldwide. And we would like to thank you for all the invited speaker, for the Professor Haripan and Dr. Anna and Professor Nurul Ulfa. Very warm thank you from us for the committee, and for giving us the very fruitful discussion and insight knowledge today. And for the next session, Professor Rohani will be a chair for the next 3 speakers. Thank you very much again for Professor Head Department, Dr. Anna. It's very nice to see you for this symposium, and we hope we can continue collaboration by doing research collaboration with you. Thank you.
Thank you very much.
You're welcome.
Thank you. Have a nice meeting.
Thank you, everybody.
Thank you, Dr. Asmicitra, for having incredibly lead the session. Before we continue to the next plenary session, we invite Dr. Asmicitra to join us in front of the stage for receiving the souvenir from the committee. For this, we invite Chairman of the Symposium, Dr. Aslamia, and Director of Aquatalk, Professor Iwanuddin, to give the souvenir. Thank you. Please be seated.
Ladies and gentlemen, the second plenary session, we have three main excellent speakers. The session will be led by Professor Rohani Amborape as moderator. To Professor Rohani, the stage is yours.
Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi. Good morning. I'm Rohani Amborape. I'm a moderator for this second plenary sessions. For these sessions, we will have three excellent speakers. Dr. Davin Setiamarda. The second one, Dr. Mukti Sainuddin, MSc. And the third speaker, Dr. Amanda Lindsay. So we will have Dr. Davin and Dr. Amanda via online platform, and we will have Dr. Mukti Sainuddin in an offline platform with us in this room. So we have, we allocate 20 minutes for presentations for each speaker. And then I would like to have all the three speakers will present their presentations, and then we continue with discussions at the end. So I would like to invite the first speaker, Dr. Davin H.S. Setyamarga. His expertise in molecular biology, and Dr. Davin is an associate professor at the National Institute of Technology, Japan, and he's also a visiting researcher at the University Museum, the University of Tokyo. He has published over 40 publications with 1,573 citations. And then he said that he's evolutionary biologist and zoologist, and he understood how animal diversity arose and evolves using molecular biology, genetics, and genomics methods. And then he worked mainly on marine and aquatic metazoans, and starting to work with terrestrial creatures too. So we will have Dr. Davin, and please, Dr. Davin, your time.
Thank you very much. Can you hear me?
Yes.
Okay, very well. I think that's the most spoken line during the last 3 years during the COVID period. Can you hear me? Because everything is done online, right? Can you hear me? Can you see me? Can you see my video? Can you see my slides, right? Those are the most used sentences during the last 3 years. Okay, thank you very much for the introduction. As introduced by the moderator, maybe should I share my slide now? Okay.
You can share your screen.
Sure. There you go. I don't think, yeah, because I think rather than look at my face, it's better to look at my slides. Alrighty. Oh, sorry. I think I did a mistake here. There you go. Okay, so I'm an associate professor of molecular biology at the National Institute of Technology, Wakayama College in Japan. It's in Southwest Japan, about 2 hours car drive from Osaka. And I'm also an adjunct research collaborator at the University Museum of the University of Tokyo. So I work mainly on metazoans, so on animals, basically. I've been working on a lot of marine animals, but I also have started to work on some terrestrial animals and even plants and also bacteria. Well, but so basically, my interest is more on evolution and environmental biology. So I've had a pretty extensive discussion with one of the committee member, asking whether I should give a talk with a more general topic, or if I should introduce the result of one of my most recent research result, which was presented yesterday at the Japanese Society for Animal Systematics. So the committee member suggested that I talk more on a specific topic to introduce my research. So please pardon me if the talk is heavily technical. Yesterday, I also attended the opening ceremony, and I think from my questions and my comments yesterday, a lot of people already realized that I'm actually not a person who work on applied sciences or who are interested in trying to make products. I'm more of a fundamental research scientist, so I work on mainly on evolution. So rather than working on conservation, I try to understand the genetic mechanisms of diversity. So if anybody else would like to use the information for to be applied on conservation, go ahead, but I'm not going to do that. So that's my stand. So today I'm going to talk about my latest research result about the Argonauts. It's a member of the cephalopods. As we all know, the cephalopods do not have any shell, but Argonauts have some secondarily acquired shells. So I try to dissect the evolution of the acquisition of the shell of the Argonauts through from the molecular biology standpoints using various multi-omics methods. So, right. So as we all know, mollusks. Everybody knows mollusks. You know, you have shells, clams, oysters, etc. One of the most defining character of mollusks is the presence of an external calcified shell, right? And then the calcification is actually based on calcium carbonate. And evolutionarily speaking, molluscan shells evolved very early during the Cambrian period. So it's already like emerged around 500 million years ago. Although the detail of the formation of the shell has not yet been elucidated completely, thus far we know that it is being formed through the secretion of proteins from the soft tissue called the mantle, and then with the mineral molecules precipitate on the proteins, and then forming layers after layers of minerals. And then this is the conchiferans. So conchiferans are a group of mollusks, as you can see here, a group of mollusks with very clear external calcified shell. Members of so you have snails, you have bivalves, you have clams. They're all scapopods. They're all scaphopods. They're all actually conchiferans. And actually, cephalopods. Sorry, I have to do this. Right. So cephalopods is actually also a member of the conchiferans. But we all know that cephalopods include squids and octopuses, and then octopuses do not have any shells. And the extant octopuses and squids do not have any shells. However, in the past, many species of sorry, I'm actually in my car doing my presentation in my car. Right. So we all know that in the past, well, although the extant members of the cephalopods mostly do not have any external or clearly calcified shell. Well, of course, they sometimes have some degenerated organs homologous to the shell, but sometimes they do not have a very clear external shell. The phylogenetically basal member of the group, like the nautilus here, actually has a very big shell, and then it's calcified, it's completely calcified, and it's also well, it's also external. So it's clearly a mollusk shell. And also, of course, one of the extinct member like the ammonites here, they're also known to have external shells. But other members, such as the octopuses, for example, they do not have any shell at all. And then members like the cuttlefishes, for example, they would have cuttle bones, slightly calcified, but it's small and completely externalized. So they're not like one of those beautifully, calcified external shells like the shells of the nautilus or other mollusks. In one of my previous papers, I conducted myself and my collaborators and my research group conducted multi-omics analysis. So what we did basically is transcriptomics and proteomics of the shell and the mantle tissue of Nautilus pompilus. So as I mentioned earlier, the Nautilus is located here. So it's basal, phylogenetically basal when compared to the rest of the octopods, to the cephalopods. And then we found out that many of the shell matrix proteins, so the proteins contained inside the shell or the proteins being used to form the shell, are actually being conserved among the conchiferans, including Nautilus. So the Nautilus shares these proteins with other conchiferans. So it's not specific only to the Nautilus. So this basically also provide another evidence that the Nautilus is a member of the conchiferans because the Nautilus have similar proteins with regards to its shells. However, there is also another member of the cephalopods called the Argonauts. The Argonauts actually also has an external shell, right? It's a member of the cephalopods and external member of the octopods. It has like this, and this is congeneric species, the Argonauta argo and Argonauta hyans. So they have calcareous shell. Well, they're calcified, but they're very brittle and very thin. And then the shells only present in female. Only the shells are present only in females. And then this is thus far nobody has actually did any molecular dissection, but in the past, about I think during the 1800s or something, a researcher observed the behavior of cell formation in this group, and then they found out that they deduced actually that the shells were formed not through the secretion by the mantle tissue, but apparently it was formed through the secretion by the arms. And then the shell is easily detached from the organism. So unlike the other molluscan shells, it is said that the function probably is to maintain neutral buoyancy and also to store eggs and to carry eggs around. So therefore, based on those observations, previous researchers think that this is probably an autapomorphy, and thus the shell is probably not homologous to other conchiferan shells. However, thus far, nobody has ever done any molecular analysis trying to figure out if this is the case or not. This is my student here. So my student and I conducted another research using full mitochondrial genome sequences and conducted phylogenomics analysis to figure out the position of the Argonauts among the octopods. We found out that the Argonauts are actually form a monophyletic group together with the blanket octopus, the species with no shell at all. So it's a shellless octopus. And of course, outside this clade, you can see the rest of the octopus here. Of course, they don't have any shell at all also. So apparently the Argonauts is very interesting because the Argonauts here, the two species of Argonauts, they have shells, external shells, but the rest of the group, the rest of the group have no shell at all. And this is the squids, of course, right? And the squids do not have any shell. And then so we would like to figure out how the Argonauts acquired their shells, and then to see if the shells of the conchiferans are actually homologous to the shell of the Argonauts by looking at the matrix proteins. So the proteins contained inside the matrix of the shell is called shell matrix proteins, while the shell of the Argonauts, because the function is to protect its shells and only formed by females, it's also called the egg case rather than the shell. So in order to differentiate the shell of the Argonauts with the shell of the conchiferans, we use the term egg case for the shell of the Argonauts. So for the matrix proteins, we also call the proteins egg case matrix proteins. First, we collected samples from the Oki Island. It's in over here. It's Oki Island is over here, so it's close to it's on the Sea of Japan. So it's South Japan, Southwest Japan on the Sea of Japan. This is called, and this wait a minute. It's better for me to this is not mixed. This is fixed net. Sorry about that. Fixed net at Oki Island. So we got samples, fresh samples, fresh animals from the fishermen. The fishermen caught the animals with the animals caught by the fixed net, and then we dissected the animals, and then we collected the first arm, the second arm, and the mental tissue, and then we did transcriptomic analysis. And with the rest of the body, we sequenced its full genome. Meanwhile, we also crushed the shells and then extracted these proteins and then conducted proteomics analysis. And then we compare the proteins, the proteomes and the transcriptomes result. And then afterward, we annotate the proteins based on the result of the genome sequence. We were able to identify 29 proteins in Argonauta argo, while we were able to identify 33 proteins in Argonauta hines. In Argonauta argo, we were able to identify functional domains in all proteins identified in the species, while for hines, we only able to identify the presence of domains in 31 of the 33 proteins identified in the species. And then when we compared the protein of the shell, the egg case matrix proteins of the two species, we found out that the two species actually shared 11 proteins. So this is the name of the proteins. Share only 11 proteins. So about the other 18, we do not know if the other 18 or the other 22 are actually species-specific proteins. But there's also a possibility that is because the data, the annotation data, were well, there are a lot of different causes we were unable to identify the 18 and 22. However, we were able to, we are sure that there are at least 11 proteins being shared between the two species. Got it? Okay. And then later, we tried to compare the obtained proteins in the Argonauts with other cephalopods. So we compared the egg case matrix proteins of the Argonauts with the shell matrix proteins of the Nautilus and the octopus genome. Of course, we know the octopus do not have any shell at all, but we tried to find the genes in the octopus genome. And then interestingly, these are our findings. First, all of the egg case matrix proteins and the shell matrix proteins are actually conserved in the octopus genomes. So we found all of the proteins in the octopus genome. So this is very interesting because that means that the octopus have all proteins, all of the matrix proteins, shell matrix proteins needed to form the shell, and all of the egg case matrix proteins needed to form the egg case. But the octopus apparently, while it has all of the tools, it forms nothing. The octopus has no shell and has no egg case. And then later, we also found that the egg case matrix proteins in the octopus genome and the shell matrix proteins of the Nautilus in the octopus genome do not overlap, which means they're not sharing proteins at all. which also means that the egg case are being formed using different set of proteins than the proteins being used to form the shell of the nautilus or other conchiferans. Of course, there is also common proteins. We also identified common proteins between the egg case matrix proteins and of the Argonauts, and then the shell matrix proteins of the Nautilus. And the proteins are actually the chitin binding domain containing proteins, which has been identified as one of the major shell matrix proteins in other conchiferans also. So to conclude this section, we could say that almost all egg case matrix proteins are present in the genome of the non-celled octopus, while at the same time, the set of the shell matrix proteins of the Nautilus populace also which among the cephalopods has retained the true external shells, is clearly different from the set of the egg case matrix proteins of the Argonauts, which means that the egg case or the shell of the Argonauts is not a conchiferan shell. Okay. Right, and then we also compare the matrix protein of the matrix proteins of the two Argonauts with other conchiferans, besides the Nautilus, and then we found no shell matrix proteins being shared among the 5 species. However, let me I'm trying to voila. Right, and then now when we are trying to look at the proteins, but as already previously mentioned, there are some proteins conserved among some of the conchiferans. So although no proteins are conserved among all conchiferans, there are some proteins conserved among some conchiferans. And then the protein is the PIF-like laminin G3 protein, which is also known as a very important shell matrix protein because this is involved in the calcification, in the formation of the aragonite or the calcium carbonate crystals of the shell. When we did phylogenetic analysis, molecular evolutionary analysis of the protein, though, we found out that the cephalopod has its own protein set of proteins compared to other the proteins of the gasteropods and bivalves. Bivalves and gasteropods shares the same protein, so they are basically homologous. However, the cephalopods apparently use a different paralog of the laminin G3 domain containing proteins. So the proteins in cephalopods are actually not homologous to the other proteins, but they're only paralogous, which is actually very interesting because when we checked our Nautilus, our Nautilus paper, we actually also found the same result. Later, we also tried to check the proteins only being found in the egg case and not in the shells of other conchiferans. One of the proteins is actually glypican, and glypican is very interesting because glypican is actually the major protein for chicken egg shell. So,
sorry, Dr. Delphine, you have 2 minutes left for your.
Okay. So basically, glypican is actually being recruited by the avians to form its eggshell independently by the recruitment of glypican by the Argonauts to form its egg case. Therefore, now we can conclude, based on our result here, we can conclude that the shell of the Argonaut is not a true shell, but an egg case, even at the molecular level, because we can see that there is no similarities. There is no similarity between the egg case matrix proteins of the Argonauts and the shell matrix proteins of the conchiferans. Meanwhile, we also suggest that the egg case formation in the Argonauts were probably acquired by ritualizing a handful of conchiferan cell matrix proteins and simultaneously by recruiting many proteins originally unrelated to shell formation, or like, for example, like glypican. These are the fundings I got to do this research. So mostly from the Japanese government or from some companies like the Asahi Glass Foundations. We also got some support from the Japanese government to do the full genome sequencing. Thank you very much. And then I dedicated my talk here to my best friend who just passed away last month. Thank you very much.
Thank you. Very interesting presentations, Dr. Davin. For the participants in our online platform, I saw here more than almost 160 participants in our online. You can put your questions via chat room for the moment while we will have other presenter to present their presentation. So please write in a chat room for your questions if you have for Dr. Davin for now. For the second presenter, I would like to invite Dr. Mukti Sainuddin. So Dr. Mukti Sainuddin, please come to the stage. Okay. I would like to introduce Dr. Mukti Sainuddin. His expertise in a fisheries information system and marine geospatial, and then he's associate professor at the Faculty of Marine Science and Fisheries, Hasanuddin University, Indonesia. He has published over 78 publications with 1,274 citations, with 3 books and 3 IPR in the last 5 years. So, Dr. Mukti Sainuddin, I invite you, your time now. Please share screen for Dr. Mukti presentation. Okay, so while waiting presentations from Pak Mukti Sainuddin, probably Dr. Mukti, could you please introduce yourself to the participants?
Yeah, okay. Thank you very much. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Okay, my name is Mukti Sainuddin from Hasanuddin University. Today, I would like to talk about the detection of ocean hotspot for pelagic species within the Indonesian Fisheries Management Area 713. I think this is we hope that this will be get important implication for fishing strategy and management. So let me explain simply about overview. My presentation we will divide into two categories. The first one will explain about a hotspot from global perspective, and also then particular perspective, especially in Indonesian fisheries management area 713. In the first one, I would like to introduce what is the hotspot and why the hotspot is very important for especially capture fisheries. And then we'll continue to data and methods. I would like to describe briefly the simple approach we use to detect the ocean hotspot. And the next, I would like to talk about the hotspot in specifically, such as prawns, whaling, eddy, and how to generate or construct potential fishing zone. And last one is concluding remarks. Well, today I will talk about how to explore or expose the hidden ocean hotspot for pelagic species, especially commercial fishes distributed from global ocean and also Indonesian water. Here are the first one. I would like to talk about the simple definition of hotspot. Hotspots will known as area of high biological productivity stimulated by physical processes and primary production, secondary primer, and also will stimulate area of high high fish production. And the hotspot also can control the fish distribution, migration, and abundance of the especially pelagic species in the ocean. Generally, the hotspot consists of such as upwelling, mesoscale eddy, and prawns. Maybe this is one of the important habitats for fishing ground all over the world. But using long-time data sets such as satellite or remote sensing, we can get opportunity to identify the resilience of pelagic marine hotspot. For example, this is cyclonic eddy can enhancing nutrient input to the surface ocean. This is also very important part of a hotspot that we can find in Indonesian waters. And next, why hotspot is very important. At least I think there is three main reasons why hotspot is very important. The first one, this area is very important for setting fishing strategy and management of fish stock. And the second one, higher probability of increasing catch productivity and business profit. And the last one, this is a tool for setting fishing conservation or conservation priority, and also play important roles in decision-making process, especially about how to exploit the pelagic resource optimally. Okay, let me describe briefly how to explore the hotspot. The first one we explore the hotspot, it's about 2004 when we have international conference in PICES, 13 PICES conference in Hawaii. We talk about especially hotspot for pelagic species, and this is the one of important meeting talking detail about ocean hotspot for pelagic species, especially tuna species. From this conference, we construct the how to define and generate model to expose ocean hotspot, for example, albacore for albacore tuna. We use the simple data set consists of surface temperature and chlorophyll concentration, and also from this data we try to develop a model and define where is the potential fishing zone for hotspot for this species. And also this result we have also published in deep sea research in 2006. And the next one, we use a statistical model by combine generalized additive model and generalized linear model. And we have three data sets such as temperature, chlorophyll concentration, and sea surface head anomaly. We combine and we take into the model, and we try to generate spatial space-time ocean hotspot for albacore tuna. And we published this paper in Fisheries Oceanography 2008. The other important things talking about hotspot, how the temperature prone associate significantly with blue whale. This is from Etnoyer 2004 in the North Pacific. We can see that the hotspot can sustain until 1 year. And we can see here how distribution of blue whale is concentrate the location of persistence of hotspot. Another fact we can see from Royer et al. 2004, using sea surface temperature and chlorophyll concentration to define oceanic prawn, and try to correlate with the bluefin tuna distribution and this prawn. And they found this is any significant contribution of how a hotspot can attract the fish school to make a potential fishing ground for tuna species. Another results we can see that from Nieto et al. 2017, we can see clearly how the hotspot generated from ocean carrying data, temperature data, also done in chlorophyll concentration. They combine in a statistical model such as bootstrap regression tree, and try to relate the relationship between albacore tuna and this prawn. One of the phenomenal or important scientific finding from Filipina et al. From 2001 they try to track the migration pattern and also the fish concentration all about Cartuna using only one oceanographic parameter. This is chlorophyll concentration from the seaweed sensor. We can see that the albacore concentration associate very well with the chlorophyll concentration front in the North Pacific. Now again, we can see one of the important part of hotspot. This is eddy field. You can see from research Seki et al. 2004. Also, we can see clearly how the eddy field can attract pelagic species such as big eye tuna concentrate near eddy field, central of eddy field. This is also one of the fact. This hotspot is very important. And how about in Indonesian water? Next, I would like to explain in more detail. Again, in our research also in North Pacific, we have also to simulate how the eddy field ocean generated from ocean current from the image of sea surface height. We can see how the albacore tuna concentration aggregate near the front and can sustain until 1 month in this location. Now, the special important information from Indonesian water we can see. This is from Atma Difora and Widya Stuti 2014 and also Gordon 2005. You can see that Indonesian water, especially in the central waters of Indonesia, we call Indonesia Fisheries Management Area. We can see from north and southeast monsoons is very different characteristic in terms of temperature and also chlorophyll concentration. During the southeast monsoon, the higher the chlorophyll concentration is very higher, and the opposite with temperature. On the contrary, in the northwest monsoon, the chlorophyll is low and temperature is tend to be high. Now, from this, this is one of our motivating questions. How about space-time hotspot in the central Indonesian water? It's about 10 years ago, we have explored this point in the central Indonesia, especially in Indonesia Fisheries Management Area 713. Why we will study more about this point in this area? Because the Indonesia Fisheries Management Area 713 contributes about 12% of total Indonesia national fish production. This is one of important reason why this area is very important to explore more about hotspot. Now, how to generate hotspot in space and time? Here, you can see that we combine fishery and satellite data into one data set. And satellite data consists of temperature, chlorophyll concentration, and ocean current, and also from fishery, fishing position, catch period effort, and also statistical data. We combine this data to define the free per oceanographic factor. Why we try to detect a free per oceanographic factor in space and time? Because this is one of important indicator to detect ocean hotspot. And also from this, we also generate temperature and chlorophyll from the seed algorithm. And also we try to apply a statistical model such as GAM, GLM, or kernel density estimate to construct mapping of the hotspot. Now, until now, we have several research about how to detect and predict and also forecasting hotspot in space and time. Because this is very important to track migration route of pelagic species, especially such as tuna and also mackerel, skate, and so on. And this is very important to for application of fishing management and strategy for fisheries resource in central Indonesian water. Here, I would like to show you how the conventional fishing in Indonesia rely on artificial fishing grounds such as fish aggregating device and also the seabirds flying or swooping in near surface and also foam over the sea. Now, from this, the conventional or traditional method, we can also develop to identification or detection of ocean hotspot. This is very helpful for fishermen for detecting the potential fishing ground. And this is our research also in 2017, how to predict skipjack tuna in Indonesian fisheries management area using GIM. And we found there is a movement of hotspot from 1 month to another month. The first one we can see, they have aggregate the hotspot developed in Makassar Strait and then move to the Flores Sea and then continue to stay in Flores Sea. This is very important to develop the fishing management because migration route of the species we can identify from time to time to detect the fish school concentration. And here I will show you how the cyclonic AD and also anticyclonic AD we also find near Makassar Strait. Here we can see how the skipjack tuna concentration also aggregate in anticyclonic AD near from Pare-Pare, maybe or Forman coastal waters. And here we would like to highlight how to combine multiple oceanographic parameters such as primary production, chlorophyll concentration, and also surface temperature. We put into the model and also we try to map out the hotspot using this is the top of map. This is the skipjack habitat index. We define habitat index based on the preferred oceanographic factor of temperature, chlorophyll, and primary production. And we can see that this is also very, they have very good correlation between hotspot habitat index and also catch data concentration. And then we also try to develop thermal prawn, and we published in 2020 in one of national paper. We can see here the spatial distribution or of thermal prawn, sea surface temperature from MODIS sensor or aqua satellite. We can see here how the skipjack tuna concentrations distribute near from the front and also develop about 1 month this area. So here also we can see the distribution of hotspot in relation to skipjack tuna fishing ground in the Flores Sea. And we can see also have a good association with chlorophyll concentration. So from here, what we learn from here, there are three kinds of hotspot we can find in Indonesian fisheries management area. The first one is oceanic front, chlorophyll, and also temperature prone, and also epiphyll, and the last one is upwelling. But I think the upwelling have indirect effect relationship with the pelagic concentration with these oceanographic features. And here I found that the distance of the skipjack concentration relative to thermal front, the fish concentration tended to be high when the distance between front is nearest. So we have examined also, this is statistically significant. And one last point I think we would like to explain, this is also we have to publish in 2017, how to explore the hotspot in Bone Gulf and Flores Sea. We can see here from May yeah, okay. Here, just I would like to show that this is the hotspot in Bone Gulf and Flores Sea also have a good correlation with fish concentration. And we have also examined statistically significant. And here I would like to show you how the hotspot in the Bone Gulf can persistent from 1 until 5 years. And we can see which area which have a high probability of finding this species, especially skipjack in this area. And the last one I would like to show you, this is how hotspot the flying fish. This is the special endemic fish in the central Indonesian waters. We have also defined where is the hotspot of this species. You can see this slide. We have defined this area, and this have a good association with ocean current, chlorophyll concentration, ocean depth also, and temperature. I think what we learn from here, the first point, it's hotspot. It's very important for commercial species in Indonesia fisheries management area. We have defined the skipjack tuna, mackerel, skate, and flying fish, the hotspot position in this area. And also this hotspot can assist and very important for developing operational fisheries oceanography through ubiquitous fisheries information system. We can also construct this system to mapping out space-time of the hotspot position along the central Indonesian area. The last one, this study provides important step, I think, for fishing management and conservation. That's all. Thank you.
Thank you very much, Dr. Mukti. So nice presentations, but we wait for the question and answer after the last speaker. Now I invite Dr. Amanda Lindsay. Hi, Amanda. How are you?
I'm good. Thank you for having me.
Sorry to keep you up late. I don't know what time is it now.
I'm lucky. It's only 10:30 p.m.
Okay, that's a good timing.
It is still good.
Yeah, so our third speaker, Dr. Amanda Lindsay, her expertise in agricultural and natural resource economics. She's now a lecturer and researcher at Luther College, United States of America. She did her PhD from University of California, Davis, And for part of her PhD study, she did it in Selayar Island. So she has learned a lot and then have some case study from our area in Selayar archipelago. And Amanda did her master degree from University of California, Santa Barbara, and bachelor degree from Colby College Environmental Science. Sorry if I'm wrong, Amanda. And
No, you right.
so Amanda is an environmental and research economist, and her research focuses on the evaluations of regulations and management of natural resources, as well as the quantifications of impacts to resource users. She has a really nice publications on a proceeding of the National Academy of Science in 2020, entitled Evaluating Sustainable Development Policies in Rural Coastal Economies. And I think most of the data is from Selayar, isn't it, Amanda?
It is.
Okay, I give a time for you, Amanda, to present your presentations, your time.
Great. So hopefully you can see my screen. Thank you for the wonderful introduction, Rohani. I am so happy to be part of this symposium. I've really some wonderful scientific research that has been presented. And while I don't have as much experience as our other speakers so far, hopefully I can add some different perspective into thinking about the way that we can achieve sustainable development objectives, particularly when we think about managing our marine ecosystems. So I don't think I need to convince anybody here, and the wonderful talk just before pointed out that these coastal fisheries, particularly in Indonesia, are so incredibly important for the entire world. It is an intensely important area for biodiversity. And not just biodiversity, it's also extremely important in terms of supporting livelihoods and economics of the region. So estimated over 120 million people depend on coral reefs for food, for income, and protection from natural disasters. And not just the people in Indonesia care about this region, but all across the world, as it's an important spawning and nursery ground for tuna industry worldwide. I think that it's very encouraging the past several years, more and more countries and leaders have been recognizing the importance of sustainable development. In particular, we see an increased focus on our management of marine resources and how that fits into addressing poverty, education, gender equality. All of these things are connected. And so it's really important that we think about the scientific management as well as the socio-economic management of these resources. Indonesia is really amazing. It's a very large contributor to global fisheries. But I think what a lot of times people don't realize is that small scale fishermen are so important to our global production of fish. Recently, the past couple of years, over half of the fish that are consumed worldwide are now coming from developing countries. And in these tropical regions, small-scale fishers play an extremely important role in the harvest, capture, and trade of fish. So what I'm going to be talking about today is thinking about what does economic theory paired with our ecological understanding, what does it say to our ability to achieve sustainable development goals? Since the 1980s, there's been a lot of debate between economists about whether or not sustainable development policies can actually work. A lot of this research is focused on what I like to call green policies. These are the traditional policies that focus on sustainable agriculture or perhaps ecotourism. I'm sure many of you know, because Indonesia is a pioneer in this new movement to promote blue sustainable development policies. So these are new types of policies and programs that focus on marine income generating activities. They focus on using these economic activities to alleviate poverty and also conserve our ocean ecosystems, because the fisheries are important. And as we've seen from earlier presenters today, it's important as a sink for carbon and other ecosystem services. So I'm a trained economist, so I think a lot about economic theory, and there is a lot of evidence out there that suggests that blue economy programs might not succeed when they are implemented in rural areas. So a very big overview. This is a very complicated literature, but some things that we need to watch out for is first, missing and imperfect local markets. So what I mean here is when there are regions which are unable to trade with bigger markets, that can create kind of local pockets where we see unexpected responses to policy driven by changes in supply and demand. We also know that households, when they are heterogeneous, when they are different from one another, it can also have these unintended consequences. So we have to be mindful when there are wealthy households and very poor households, when households have different access to alternatives, they can be affected by these marine policies in important ways. Economists in general know that marine resources are very unique. Many times the resources, the fisheries themselves, are open access, or they're managed by local groups or resource users. And that means that it's a little bit harder to predict how fishermen are going to respond to management. It's perhaps a little bit more difficult than, say, studying farmers who own the land that they're working on. Recent work, and these are some of my colleagues who do similar style research as they have been kind of documenting and showing how marine resources are particularly complex. And so even if you do a policy that is only a few years in length, it can have long-term impacts because our fisheries will continue to grow and change responding to disturbance. And so in the next series of slides, I'm going to kind of give you a representation to show you how my modeling framework operates. So we want to think about a fishing household who perhaps is only accessing nearshore fisheries in coral reefs or seagrass. This is considered maybe not sustainable, and perhaps policymakers want to encourage these households to fish from a more sustainable fish stock, such as our offshore fisheries. So a common policy would be to provide these poor fishing households with gear that allows them to access these offshore fisheries. And many times we'll have marine fisheries researchers study how this household is making their decisions and how this policy impacts their harvesting. My work, however, stresses the importance of thinking about the bigger economy and how this household fits in amongst its neighbors. We have non-poor fishing households that compete for similar resources, they will be indirectly affected by this policy. Even our non-fishing households, so these are households who are engaged in agriculture or other businesses, they are going to be impacted by this policy because they'll see changes in the price of fish. Maybe wealthier households will now demand more goods and services. And it's important that we think about how a simple policy like providing a household a boat could have these indirect impacts. And of course, as the fish stock either shrinks or grows in response to new fishing behavior, we need to quantify those impacts as well. And so what you see here is a representation of my general modeling framework. I use economic theory from development economics and natural resource economics and collaboration with ecologists to create a model that we call BioLouis. This is a local economy model, which has the different household groups. We modeled the different economic sectors, the supply and demand of these goods and services, and also the important biological properties of local fisheries. So I've really enjoyed my time in Indonesia and can't wait to return, but Indonesia is a fantastic place for me to focus my research for a lot of reasons. Indonesia is a very important economy globally. It is making massive gains the past decade in poverty reduction nationally. And it's also, I think the last statistic I found said it's the second largest producer of marine capture fish in the world. You guys produce more fish than the United States. And as you know, a lot of this fish production is happening in Eastern Indonesia. Your fishing fleet is dominated by small fishing vessels. And I mean, Indonesia has so many islands and there's limited infrastructure, which means that all of these communities from which we are obtaining these important fish resources, there's a lot of potential for these areas to be isolated. And so it's important to think about these economic linkages when we are considering how to more sustainably manage marine fish stocks. As Ibu Rohani mentioned, my data collection for this research came from Selayar in 2016. I worked with a group of current and former UNHAS students, and we collected almost 500 household surveys and over 250 business surveys throughout the island. Very importantly, there's a mix of industries that the households engage in. The enterprises and businesses were very small, on average, less than 2 employees per business. So it's really important to think about household producers and how they have constraints that affect their ability to generate income and support their households. I don't want to go into too much of the mathematical detail because I don't want to scare anyone away, because I think this research is really important and valuable. But let me just talk about a few of the big features of our model. The model has this general equilibrium component. This is just a fancy economic term we use to talk about finding the place where supply equals demand. And basically is the cornerstone of economic theory. The main industries in our model include agricultural production. For marine fisheries, we separately model the production of nearshore fisheries from coral reef and seagrass, from offshore fisheries, so that's like the skipjack and tuna and mackerel, and also some other small household enterprises. We model the different groups of households. So we model these different components based on household income in relation to the national poverty line and whether or not they engage in fishing. And while there's a lot of assumptions in the model, it's important to note that the nearshore fish price and the labor wage, so this is your hourly wage rate, these things are determined locally. And what that means is that when the supply or the demand for either nearshore fish or labor changes, we're going to see a change in the price. And that's going to be really important to remember when we're looking at some of our results. The second component, perhaps people in the audience will feel more comfortable with this part of the model. We use a delayed difference biological model to represent our nearshore fish stock. Generally, this means that we are accounting for natural survival and growth, as well as harvest from local fishermen. Our modeling framework simulates policy impacts for a 10-year time horizon. And after that, I think our assumptions start to break down. So we like to be a little bit more conservative with our simulation results. What I'm going to be presenting right now quickly is some of the results and findings that we have from this recent publication, where we looked at a combination of different policies that could achieve sustainable development. And then if I have time, I'd like to say a few words about some exciting work I'm working on right now. So the policies that we are thinking about is providing these larger fishing vessels that can be used or more easily used to capture offshore pelagic fish. Also increasing fishing capital restrictions. So could we implement a policy where we make sure these larger boats stay further from shore and are not used close to the shoreline? And agricultural capital provisions. I included this policy as a more traditional type of sustainable development policy in which households are provided resources to improve the value of their agricultural production. We simulate these policies with our economic model, and to evaluate them, we look at how the nearshore fish biomass responds to the policies. And we also importantly look at how incomes of households are affected after these policies are introduced. I'm just going to highlight some of the big picture results, and I hope that this gives you an understanding of how the model works and what kind of value we can get from using economic and ecological sciences together. So the first simulation was when we provided a fishing vessel to some of the poor fishing households. In this policy, it was equivalent to providing 45 gross ton fishing vessels to poor fishing households. When that occurs, these households become more productive. And because they are more productive, we expect the local wage rate in the economy to increase. This means our poor fishing households become wealthier, but also many other households become wealthier. And this increased income means that households are demanding more goods. They're demanding more retail goods, more restaurants. They'll also demand more fish. This puts pressure on our local prices, and the higher price of fish is expected to incentivize all fishing households to increase the amount of nearshore fishing that occurs. So here's a way to visualize the results. We have a percent change in biomass in blue, in labor in red, and capital in orange. And what this results show is that even though only the poor fishing households are receiving the offshore fishing vessels, this higher price of fish is encouraging all households to increase the amount of effort in the nearshore fishery. And so we would expect the nearshore biomass to actually decrease over time. When you look at these results, it's a little bit alarming. So you might think, okay, what if we have the vessel provision, but we also stop households from moving fishing capital into the nearshore fishery? So we're going to provide boats to poor households, and we are going to stop them from using these large boats in these nearshore habitats. That seems like a really good idea. However, even though our fishing households cannot move this capital, they can still increase the amount of time they spend fishing. So we see that increase in price because demand has increased and households are spending more time fishing in the coral reef, and this causes the biomass to decline. It's not as big of a decline as our first policy, but it is still a decline nonetheless. In this paper, I like to be a little optimistic. So I show a policy that seemed to help improve our nearshore biomass. In this setting, it was the agricultural capital provision. So basically what's happening here is that even though we still see that increase in the local price of fish, we've stopped fishing households from moving capital, and because we've supported this alternative livelihood, agriculture, there are this high demand for labor in the agricultural sector, and that prevents an increase in harvests, at least temporarily, allowing the fish stock to recover. So the results that I showed you were very stylized. This is for Selayar, and that might not be a perfect model for other areas in Indonesia. What's wonderful about this type of analysis is we can use the structure and modify our key assumptions so we can estimate policy impacts for other economies. Here's an example. I have changed in these graphs the labor supply elasticity. And so what that means is that in Selayar, it's very isolated and there's not a lot of people coming in for the day for work and then leaving in the evening. There's not a lot of migrant labor. But what if we imagined an economy that had migrants able to come in for a day of work and then could return home? In those types of economies, we'd have an elastic labor supply. And so while this was our preferred model for Selayar, we could imagine a different economy. And we can see that when the labor supply is elastic, all of these policies will lead to a decline in the nearshore biomass. So it's really important that we think carefully about what the particular market conditions are when we're trying to understand if these different policies can achieve our sustainable development goals. I want to spend just a few minutes if I can, but Rohani, interrupt me if I'm going too long. I want to talk a little bit about an alternative type of policy, marine reserves. We know that they are becoming really popular as countries all over the world try to protect 30% of our marine ecosystems by 2030. And what I hope to do is to think on a more micro scale what these impacts of marine reserves could be. For this research, I'm modifying that original model that I talked about. However, I'm adding a spatial component. The household surveys that we collected in 2016, we asked information about where the households were fishing. And so you can see this map here shows a heat map with these darker green areas being the more heavily fished parts of the marine habitat. We're also thinking about households that are different. So you have northern households, southern households, and these households are able to fish from these different areas. Working with ecologists, I've learned about ways to more accurately model larval dispersal, so we can think about how a reserve in one area might affect the biology and the economics in other regions. So as an example of what this type of model can teach us, I wanted to show you the results from introducing a 5 kilometer squared reserve to that first patch, that north western patch. And when you introduce this reserve, you reduce the amount of fishable biomass in the area. This graph shows us how we should expect the biomass in each of these areas to change over time. This policy leads to a rapid increase in biomass in patch 1, but there are no biological spillovers. So even though there's larval dispersal between these patch, this recovery of this area is not enough to increase biomass in other regions. That might be okay because system-wide, we expect an increase in biomass around the island. We can use this modeling framework to not only think about the environmental impact, but the economic impact. And from this graph, you can see a change in harvest for these different fishing households. Our northern households are the fishermen who predominantly fish in that area, patch 1. And so at first, they see huge declines in their harvest, but quickly their harvest levels increase because of the recovered fish stock. Again, the point of this talk is to kind of talk about the importance of market structure. And so I wanted to show you side by side. Here's my preferred specification model results. And on the right-hand side, I've changed the market structure to have unrestricted fish trade. This means there's more trade with regional markets and the price of fish is not sensitive to changes in supply from local fishermen. And what you can see here is that we have a much larger increase in biomass in patch 1, and we do not experience a decline in biomass in these other regions. This suggests that a marine reserve will be more successful in increasing biomass, sorry, when fish trade is less restricted. My hope is that, sorry about that. My hope is that you understand that market structure is very important. This type of modeling methods is very technical, but I think it is very helpful in providing policy analysis. This methodology, while there is a lot of mathematics involved, it can be used in data poor settings where we don't have a lot of record keeping on fish harvests. And importantly, it can be used to help predict how policies will operate in other similar regions. I am not the only researcher involved in this type of work. We're seeing an increasing amount of research that is taking this interdisciplinary approach to understand how both economies, people, and ecosystems will respond to changes in tourism, in changes in regulation of marine resources, and other types of sustainable development policies. Thank you so much for having me.
Thank you, Amanda. Sorry, maybe because you need to, stay up night. So
no, I think I'm because I'm recovering from COVID. I still have a bad cough sometimes.
Yeah. Sorry for that. Okay. So we just listened to three
speakers' presentations, very nice with a quite wide range of the background. We have molecular biologists, we have fisheries information system, and then we have from natural resource economics. So really nice range of the backgrounds with the same objectives to how to think to sustainably manage our marine and fisheries resources. So I invite the questions from the participants, either from this room or from the online platform. Actually, we have already questions from the chat room. This is to Dr. Dalvin from Pak Sulistiono, IPB. Dr. Dalvin, you still with us? Hello?
Yes.
Okay. Yeah, so this is questions for you. Asking about the study on mollusks in Indonesia is very limited and not many research on these topics. And then Pak Sulistiono asking if there might be certain seasons or times when a student research certain biota. And then how are mollusk research activities in Japan, especially at the University of Tokyo, and are there special scholarship or special research funds that students can access? And can a similar program be applied in Indonesia? And what topics are interesting enough to collaborate?
Okay, so I will try to answer based on my capacity. Please understand that my home institution is actually at National Institute of Technology, Wakayama College, and it's not in the University of Tokyo. So I do not have the information or capacity to answer about how to enter other university. So, but one thing that I can say is that there is our college already has an agreement with IPB University, and some of our students already went to IPB, and I don't think there is any IPB student who already came over to our college. But yeah, well, because my college is basically an engineering college and it's only for undergrad, you could come over and do some experiments to do some research visit in my lab, for example. As for mollusks in Indonesia, yeah, well, actually mollusks research in Indonesia. In Japan, there is a very big group, and I think also in Indonesia, a very big group of mollusks collectors, and then like the Society for Malacology in Japan, well, I almost said politically incorrect word, well, basically many members in the Society for Malacology in Japan are actually amateur collectors. So, and then they do presentation, do a lot of research. And I think Indonesia, there are also a lot of shell collectors, malacologists, amateur malacologists. So I wouldn't say that research in Indonesia it's not really a lot, but probably it's not centralized. And also the other thing is that you have to understand that the research I did was very basic and very fundamental, and then so maybe the research being done by amateur or private malacologists would basically focus on taxonomy, diversity analysis, rather than fisheries or how to fatten your shellfish or something like that, So Yeah. So I think Indonesia, well, there are about 35,000 species of mollusks, only of gastropods, for example. And then probably Indonesia will have like, I think it's around 7,000 to 10,000, I think, in Indonesia only. So yeah, there are still a lot of things to be done to study molluscan research in Indonesia. But we should try to also I would say gather also the amateur malacologists and let's see what they have to say. As for what kind of research you could do or would be interested in Japan, well, I mean, I would say that, what do you want to do? First, I think if there's anything interesting that you'd like to do, and then you think it could be done collaboratively with Japan or with the US or with other countries, Singapore, Malaysia, then probably you should try to bring the idea first, rather than asking what they would like, what we'd like to do. Because of course, this is also a problem, because sometimes many of researchers in Japan or other countries would really like to get the biodiversity resources in our country, because it's very, very rich. And of course, we have our own agenda, we have our own research plan, which sometimes do not necessarily meet the interests of Indonesians or Indonesia. So probably if you have any topic of interest, you can come over to us and we can discuss about what kind of possible topics that we can do together collaboratively, so it become a win-win research collaboration, I think. As for scholarships, there are a lot of scholarships that you can apply to Japan, Mombusho scholarships, and even for research visits only for 1 month to 3 months to even a year, or like me, I ended up staying in Japan for more than 20 years now, longer in Japan than my stay in Indonesia. There are a lot of different programs that you can actually get, and I think many scholarships, the Japanese scholarships is not, like something that the information about scholarships is not closed. So it's not that you need an insider in order to get information about scholarships. You can actually get information from the embassy very easily. So I would suggest that you consult the homepage or the of the Japanese embassy, or directly to the embassy of Japan. Thank you very much. I hope that answers. Thank you very much.
Yeah, thank you, Dr. Dalvin. One questions in the chat also asking about how about nudibranchs, because nudibranchs, they said that has probably not true shell also.
Right. Yeah, I read the question. This is very interesting. I would actually also, like, I heard about this, but I don't think anybody has already researched this, in depth, molecularly. So probably if you're interested, maybe you can collaborate with me and then do research together with me. Probably if you can get some nudibranch eggs, for example, and then we can follow the developmental stages of the nudibranch. And maybe we can try to dissect the nudibranch embryos and see when calcification starts during the development of the embryo. And from there, we can probably try to, I don't know if the technology is already there. Probably we can try to see what kind of proteins or genes, during the early development of nudibranch, and then probably we can also see why they disappear. Probably it's because the gene was not expressed anymore and stuff like that. Well, but yeah, it's a very interesting research topic. Probably we can collaborate. Thank you very much.
Thank you, Dr. Dalvin. So if we still have questions from this room, Pak Wasir. Okay. Please address your questions to who?
Okay, thank you. I would like to thank about the question for Dr. Mukti. As you know about the chlorophyll concentration and SST, especially for the southeast monsoon, but I think it's not only influence about the southeast monsoon, but it's also signal about that. This is a parameter oceanography or atmospheric, local or regional. How about that? Thank you.
Yeah, thank you very much, Mr. Wasir Samad. Yeah, I think it's very interesting information about El Nino Southern oscillations or global climate change. How to consider these parameters in detecting a hotspot. Yeah, based on our research, for example, if we increase such as temperature, for example, 0.5, 1 degree centigrade, or 1.5 degree centigrade, I think the hotspot positions, especially potential fishing zone, will also move. And also, I think the potential fishing zone will reduce until about 10 or 25%. If we increase, we try to simulate some oceanographic variables such as temperatures. When we try to increase the temperatures, the potential fishing grounds also will move spatially. So I think, but during our research for last 10 years, we found that there is no significant increase in temperature and can directly affect the fish distribution. So we try to explore more detail about how the global the climate change, such as ENSO, El Nino Southern Oscillations, or Indian Ocean defaults, affect the fish distribution in central Indonesian water. But I didn't find until now significantly affect. Maybe in tropical area is just get a little effect to the fish distribution, the change of the temperature for this fish distribution. But last day we just use from 5 to 10 years data set. Then and we find the highest anomaly occur in around Upwelling area in southwestern Sulawesi Island, near Takalar's coastal waters. This is very high anomaly, either in temperature or chlorophyll concentration. What's interesting point, I think, near from Parepare and Polewali Mandar coastal area, this is very high standard deviation in temperature and also chlorophyll concentration. And we try to investigate more detail, but I think this is also maybe any correlation to geomorphology or topographic features in this area. And interesting point also, we also found some cyclonic or anticyclonic AD near that area. So in the southern side of Sulawesi Island, we found a Upwelling effect to fish distribution, but I think this is indirect effect. There is a time lag, such as maybe 1 or 3 months after Upwelling, and also fish abundance will be high at that time. And but the cyclonic eddy and anticyclonic eddy in the central Makassar Strait also, I think, have a good point for detecting this the pelagic fish distribution. But for the change of temperature, I think until now we have found a significant effect for the change of temperature and also chlorophyll, not chlorophyll concentration, also temperature. Yeah, I think so.
Thank you, Pak Mukti. Still questions for the speakers from the forum, but because in a chat room I didn't see any, so I would like to ask my questions to Amanda. That's very nice presentation about, because Amanda and her groups have developed bioeconomic models for fisheries management. And then from the presentations, with the Selayar Archipelago case, Selayar Archipelago, they have a big fishing communities, but they also have alternative livelihood in agriculture. And we already see the result for the Selayar. Do you have, analyze other fishing communities like that really just depend on a fishing, how the models could work? Thank you, Amanda.
Thank you, Ibu Rohani. That's a good question. I think that for maybe the other areas of the Selayar district where there is much less agriculture and much more fishing, it would require its own proper study. All of the research that I know of to date has these communities where there is a large mix in livelihood options. I would imagine that communities that are almost entirely composed of fishermen would have a large amount of trade in order to access those other resources that they cannot produce themselves. And so I would suspect that maybe in those communities, in addition to having other types of goods coming in from other markets, they're able to trade fish with other regional markets, which would make the price of fish much more stable. In those types of economies, I think our predictions of how policies will impact local fish stocks will be much more as expected, and you won't have these unintended consequences. I will take a moment. There was another question in the chat for me, which I didn't have time to respond to, asking about ways to sustain local fishermen livelihoods at the early stages of creating a marine reserve. And so I think that it is an extremely important point. I think marine reserves are amazing conservation tools. However, they do displace fishermen, which can have severe economic consequences. In my modeling framework, I did not find evidence of households that were 100% dependent on a particular fishing area. So maybe households were able to engage in outside labor, in agricultural production, or maybe fished in different areas. So in my setting, introducing a marine reserve maybe increases the costs for a household. It is inconvenient for a little while, but as you saw in those results, they can quickly see benefits from that temporary displacement. And then they can increase their fishing perhaps in 2 or 3 years after reserve is created. I think the point though that you're making is very important. And it's important to think about alternative livelihoods. What are other options for these households? Because displacing households for a lot of political reasons is not an option, right? We have to make sure there are ways for people to support their households.
Thank you. So Amanda, you already answered my questions and Pak Hussein Rifai questions. Well done. Okay, so I think we need to conclude these sessions and finish. So we really thank you, Dr. Davin, Dr. Mukti Sainuddin, Dr. Amanda Lindsay, for your time. So you really support our university, and then we really appreciate that. And then I'm sorry because we just able to send a token of appreciations, a token of appreciation in terms of certificates from the UNHAS rector to your email soon. Okay. So as a moderator, I would like to finish this plenary session too, and say thank you to the speakers and all the participants to attend these plenary sessions. Thank you so much.
Okay, thank you for Hani for have incredibly lead the session. Before we continue to the next agenda, we invite Prof. Hani, Dr. Mukti Zainuddin, and Dr. Nurul Ulfa Karim, represented by Professor Iwanuddin from UMT, to join us in front of the stage for receiving the souvenir from the committee. For this, we invite Chairman of the Symposium, Dr. Aslamia, Vice Dean Professor Haris, Ahmad Faisal, Dr. Aisyah Farhum, Professor Yusinta, and the Dean, Bapak Safruddin, to go up on stage to give the souvenir. To Dr. Aslamia and Prof. Haris, we invite you to give souvenir to Dr. Mukti Zainuddin. Yeah. Dr. Aslamia and Prof. Haris give souvenir to Dr. Mukti. Okay. Next to Bapak Safruddin and Prof. Yusinta, we invite you to give souvenir to Professor Iwanuddin. Next, Dr. Aisha Farum and Bapak Ahmad Faisal, we invite you to give souvenir to Professor Rohani. Next, Pak Faisal. Thank you, Pak Fani. We invite again Professor Iwanuddin from UMT to join us in front of the stage for receiving the souvenir from the Dean, Bapak Safruddin. We invite all participants to a photo session, please.
Session in the main room for 1 hour during lunch break.
Ladies and gentlemen, as we are now near to the closing of this event, we sincerely thank you for your kind attendance and participation during this session. We hope to see you again in International Symposium on Marine and Fisheries next year.
Presenters and non-presenters who have registered as participants in the symposium will be given an e-certificate by the committee, which will be sent via email.
Thank you for participation on this event. Hopefully this event will be significantly beneficial for all of us. As master of ceremony, we represent the committee, apologize for any mistakes during this event. Wassalamualaikum warahmatullahi wabarakatuh.
started. Ladies and gentlemen, all of the participants and the presenter, you can enjoy the meal, the lunch time for lunch time, while see the poster session, and we will continue the parallel session at 1:30 p.m.
Sometimes there's no one else who loves you like you used to. Sometimes we're not ourselves. There's no one I can turn to. And I know all the feelings that we shared out in the past. And I know your relationships are never meant to last. And I'll take some time, but I'll be alright. Moving on, moving on. I know times are tough, but I've had enough. Moving on, moving on,